Introduction. The Bible claims to be the history book of the universe. People have always challenged the book of Genesis by claiming it is not real history, and it should not be taken as literal history. The account of origins in Genesis is constantly under attack. Is Genesis real history? It is penned as a historical narrative. And the way the first book of the Bible is written is indicative of literal history. There is not even a hint of ape to man evolution. The account of human origins is clearly an account of special creation. God created humans separate from the rest of the biological world. There is no evidence from Genesis that humans are related to chimpanzees. Genesis 1:26. And God said, "Let us make man in our image, after our likeness, and let them have dominion over the fish of the sea, and over the fowl of the air, and over the cattle, and over all the earth, and over every creeping thing that creepeth upon the earth." Genesis 2:7. And the Lord God formed man of the dust of the ground, and breathed into his nostrils the breath of life, and man became a living soul. Genesis 2:27. And the rib which the Lord God had taken from man made he a woman, and brought her unto the man. If Genesis is literal history, then that means Adam and Eve were real people, and the global flood of Noah was a real event. This would also suggest that the earth and the universe are only thousands of years old and not billions of years old, as the secularists would have us believe. For example, in the New Testament, Peter refers to the global flood and alludes to it as a real historical event. Peter looks to the great worldwide flood as a foreshadowing of the coming judgment by fire. He makes an amazing prophecy that tells us people in the last days will deny the Genesis flood and the coming judgment. 2 Peter 3, verses 3-7. Knowing this first, that there shall come in the last days scoffers, walking after their own lusts, and saying, "Where is the promise of his coming? For since the fathers fell asleep, all things continue as they were from the beginning of the creation. For this they willingly are ignorant of, that by the word of God the heavens were of old, and the earth standing out of the water and in the water, whereby the world that then was, being overflowed with water, perished. The heavens and the earth, which are now, by the same word, are kept in store, reserved unto fire against the day of judgment and perdition of ungodly men." It is incredibly important that Genesis be understood as literal history, because it is in Genesis where we learn that death is the penalty for sin. Death came into the world because of Adam's sin. This is exactly why we have death, decay, and suffering in the world today. Romans 5:12. Wherefore, as by one man sin entered into the world, and death by sin, and so death passed upon all men, for that all have sinned. If Genesis is not real history, then why should we consider the rest of the Bible as truth? Why did Jesus have to die on the cross if Adam was not a literal man? The gospel message goes right back to the literal history of Genesis. Mark 10:6. But from the beginning of the creation, God made them male and female. In this short handbook, on independent origins, you will be presented with overwhelming evidence confirming the truthfulness of Genesis as literal history. Since Genesis claims to be the history book of the universe, we should hypothetically be able to test its claims to modern scientific data. We have done just that, and the results are fascinating. We have literally discovered Adam and Eve in our genetics. We have indisputable scientific data verifying the special creation of Adam and Eve, and conclusive evidence demonstrating that universal common descent is not scientifically plausible. This book will also provide overwhelming evidence for the global flood of Noah. Lastly, this book was written as a straightforward and uncomplicated read that is packed with tons of great arguments for independent origins and against universal common ancestry. I have written and co-written numerous books, but this specific book was meant to be that go-to book for those interested in creation and evolution, where they can find quick answers to some of the most common questions pertaining to this topic. Therefore, if you are looking for comprehensive rebuttals to all of the most common objections to the biblical model of ancestry, then please see the many other books written by teams standing for truth, especially the books specifically dealing with the critics and their objections, refuting the critics of independent origins and special creation. Also, don't forget to see Team Standing for Truth's book, refuting the critics on their objections to the global flood. Enjoy this book and make sure it is kept handy. That way you can refer to it anytime you need to for a quick and informative answer, or a strong argument pertaining to independent origins when it is required. All scripture taken from the King James Bible. That concludes the introduction. Chapter 1: Abiogenesis and Naturalism, a complete failure. Does all life ultimately share common ancestry? What is best supported by the evidence? Universal common ancestry or independent origins? What model has the best explanatory power? These are all incredibly significant questions. I have written numerous books answering these specific questions. I have also co-authored many books pertaining to the creation and evolution controversy. I am here to tell you confidently that the evidence is overwhelmingly in favor of independent origins. First, the naturalistic explanation for the origin of life has been a complete failure. If the reader is familiar with my previous books, you would know that I strongly defend the design diversity hypothesis. This model suggests that God would have front-loaded living organisms in the beginning with design diversity. This model leads to numerous accurate testable predictions and retrodictions. With simple pre-existing heterozygosity, a state of DNA differences and DNA variety, incredible variation can result. For example, with pre-existing DNA differences, Adam and Eve would easily have the potential within themselves to explain all the ethno-linguistic differences we see today. Even today, we can produce amazing variety in a single generation. We may not have as much pre-existing genetic diversity as Adam and Eve had, but we do have built-in genetic diversity. A husband and a wife could hypothetically have millions of children, and every single child would be different. This is because we are heterozygous. More on this topic later in the book. It is important to note that when the critic willfully rejects the design diversity hypothesis, they are not realizing that the naturalist has no real explanation for the origin of life. The naturalist requires the evolution of a simple, self-reproducing cell before they can even begin discussing the so-called evidence for evolution and universal common ancestry. For example, if all the athletes or the runners die at the starting line of a race, there will not be a race to discuss. The race would be over. We know that there exists chicken and egg problem after chicken and egg problem when it comes to first life. The naturalist purports that all living things came from a single cell, and that single cell came from something like a primordial soup. Is the origin of life from non-living chemicals scientifically plausible? The real truth is that empirical science has demonstrated sufficiently that abiogenesis, the origin of life from non-living matter, is highly implausible. The most common scenario for the naturalistic origin of life is called the RNA world. Here they imagine an ocean full of RNA molecules that learn to reproduce themselves and slowly evolve into the first reproducing organism. Is this a final straw in the origin of life research? It is no surprise that the RNA world hypothesis is a complete failure. Life coming from a primordial soup is a catastrophe, given the fact that water drives reactions necessary for a naturalistic origin of life in the wrong direction. We know that RNA is more unstable than DNA. In our cells, there are about 1 million DNA breaks every day, and most of these are fixed by the amazingly intricate repair systems in the cell. But if RNA is less stable than DNA, and DNA requires many amazing DNA repair systems in order to survive over time, how on earth can the imagined RNA world be plausible? As you can see, if the naturalists really wanted to make a protein from amino acids, the last thing they should want is water. I will say this again, given how detrimental it is for abiogenesis. Water drives the reverse reaction necessary for the type of origin of life scenario the naturalist wants to hope happened in the very distant past. We know that enzymes are needed as a catalyst in the cell. RNA can do this to an extremely limited extent. The reality is that in living cells, protein-based enzymes do most of the work. How did life change from RNA enzymes, from which there is only limited support for, to these amazing protein enzymes? When and how did this occur? It is science fiction to say that the first living cell evolved from non-living chemicals. There is nothing empirical about any origin of life scenario that the naturalist can and has invoked. In the book of Genesis, God tells us that everything was created very good. When we look at the modern scientific data, we see a reflection of this. Everything must have started very good right from the beginning. The incredibly complex information storage system of life could not have been built up from inadequacy to precision. If we did not have a highly functioning decoding system, the entire system would come crashing down. DNA breaks down incredibly quickly. It is extremely vulnerable to regular chemistry. Scientists know that living organisms must have amazingly complex repair machinery in order to counterbalance the chemical damage that living organisms suffer every day. For example, in a single day, we have millions of DNA breaks in our cells that are repaired by extraordinarily complex DNA machines. If we did not have these amazing chemical repair machines, we would be highly mutated. We could not survive without these forward-thinking mechanisms in our genomes. I mentioned how RNA is even more unstable than DNA. Again, this is why the imagined story of RNA arising in a primordial soup, given how unstable it is, becomes wishful thinking and requires incredible storytelling. This does not make any chemical sense. The building blocks themselves are unbelievably unstable. RNA world theorists have problems right from the start. This is why RNA world experiments are highly controlled, highly manipulated, and unrealistic. Origin of life researchers do a lot of hand waving. It is very difficult to find a real empirical scientific answer from the proponents of abiogenesis to the massive number of chicken and egg problems associated with origins research. The final blow to a naturalistic origin of life comes down to information. How do you introduce functional information into random chemicals? Even if you manage to get the necessary building blocks for life, how is information introduced? This is a major problem for abiogenesis. If I were to spill ink on paper, I would not at the same time introduce information. A book is more than just ink on paper. A book contains specified information. Let us look at another unsolvable chicken and egg problem associated with abiogenesis. Take the amazingly complex information storage system of living organisms. Even the simplest of living cells, for example, the mycoplasma, has 500 letters of DNA information. These letters and this information codes for proteins. But the problem for the naturalist arises when you cannot decode the information in DNA unless you have highly complex decoding machinery. DNA codes for its own coding machinery. DNA has the instructions to build its own coding machinery. Tell me, naturalist, which came first, the machinery? But how do you get the machinery without the instructions? Or did the instructions come first? But wait a minute, you cannot read those instructions unless you have the machinery to read it. You need everything working together right from the start. Otherwise, life would not and could not exist in the first place. This is just another one of the many unfathomable chicken and egg problems naturalists face within all types of origin of life hypotheses. I could spend the entire book covering just some of the major problems associated with abiogenesis and naturalistic origins. But I think it's already clear that evolutionists have no explanation for the origin of first life. They lack evidence and they lack any testable predictions. But they do have plenty of faith. The design diversity hypothesis, on the other hand, is empirical. And it leads to accurate testable predictions and retrodictions. The evolutionist has already failed miserably. I would also like to point out that I have personally interviewed numerous PhD scientists, including PhD chemists, who have all studied this in great detail. And they all point out just how utterly impossible abiogenesis is. Chapter 2: Universal Ancestry versus Separate Ancestry. Proponents of universal common ancestry typically look to the same, frequently repeated lines of evidence for support. The first line of evidence they could argue from would be homology. Similar structures seen in the biological world that evolutionists would say were inherited from a common ancestor. This can be applied to homology in anatomy and even homology in embryology. There exists homology in the genetic realm. Similarities exist in the biological world. Nobody would argue against this fact. Defenders of universal common descent may also argue for their model by looking to the rare so-called transitional forms, Tiktaalik, mammal-like reptiles. These would be those types of animals that appear to blend the features of different varieties of animals. Lastly, they could look to the nested hierarchical patterns, those groups within groups patterns that exist in the biological world. This is the observation that humans are more like the great apes than they are to the old world monkeys. And both humans and the old world monkeys are more similar to each other than either are to a dog or a fish. This is the structure of life. We can classify and categorize creatures in these groups within groups patterns that evolutionists assume is the result of descent with modification and ultimately universal common ancestry. Humans are grouped with primates, which is basically because we share much more in terms of anatomy, morphology, physiology, and genetics with them than we do with fish. And yet, we are still grouped with fish because we share more with fish than we do with insects, such as spiders and butterflies. Proponents of evolution would say that the amount or degree of similarity between creatures is a reflection of time since their last common ancestor. We can also look to the genetic sequences of species and see nested hierarchical patterns. Do all these lines of evidence indicate a process of descent with modification and universal common ancestry? For one, using these types of arguments to support universal common ancestry would be like looking to the sky and pointing out the fact that it is blue to argue for evolution. This would be ridiculous, since other models can also explain why the sky is blue or why the earth is round. The blueness of the sky or the roundness of the earth can both be explained by evolution or creation. Let us start with homology. When it comes to homology, we know that human designers build in homologous patterns. And we also see homologous patterns at the blueprint level. The evidence is overwhelming that what we are looking at is most consistent with a common designer. Proponents of universal common ancestry assume that because there are similarities between creatures, then ultimately all life is related through common ancestry. We find countless similarities between all the different types of cars in the world. What about redundancy? Why do cars all have a spare tire? In case you get a flat tire, you have a backup ready to go. This is all great design. It all comes down to forward thinking, which points us back to a forward thinker. Oftentimes, the earliest stages of different types of modes of transportation, sedans, SUVs, vans, all look very similar, And it is not until the final product that we see the most differences. Nobody argues against similarity. As I iterated, these shared patterns exist at the embryological and developmental stages. We see shared patterns in the early developmental stages of various forms of life. For example, we could look at a large diversity of creatures, including turtles, chickens, and even fish. And what we find are very similar patterns and similar features during these developmental stages. I will again note that it is not until the later stages where we see vast differences than during the earlier stages of development. We see this same thing in the design world as well. If you were to visit a diversity of vehicle factories from Toyota to Chevy to Volkswagen, you would find numerous similarities in the early stages of manufacturing. You would find shared patterns in blueprints, but the final products are where we would find the largest differences. What about transitional forms? Well, it is also a fact that humans build vehicles that seem to blend the features of multiple types of vehicles. One such vehicle that comes to mind is an amphibious assault vehicle. This military vehicle just so happens to blend the features of both a land vehicle and a vehicle built for the sea. This is all good design, and it is also good forward thinking. Additionally, with the invention of crossover vehicles, it has become even more difficult to classify vehicles. To the evolutionists, a crossover vehicle such as a Dodge Journey or a Ford Flex, and even the Jeep Gladiator, would be the perfect examples of a transitional form. We are just in the infancy of recognizing design. God, through his infinite knowledge and wisdom, knew exactly what good design would be. We as humans are just now realizing that transitional-like vehicles are exceptional designs. Humans have always grouped and organized things based on similarities and based on function or purpose. For example, we group tools in specific categories, and typically this is a very easy thing to do. But sometimes we come across a tool that is difficult to group, as it seems to blend the features of many different types of tools. An example of a difficult to categorize tool are fencing pliers. To the evolutionists, the fencing pliers would be considered the perfect transitional tool. The evolutionists could even line a variety of tools up in terms of similarity and function and place the fencing pliers in a spot that would give the appearance of transition and evolution. When we look to animals today, we have 5 basic groups. We have mammals, birds, reptiles, amphibians, and fish. A wrong assumption would be that we should be able to easily take fossil groups and place them in these categories. Even biblical creationists are sometimes guilty of thinking in terms of uniformitarianism. The present is the key to the past. Just because it is a lot easier to group animals today does not necessarily mean it would be the case in the past. Now, of course, we find a multitude of extinct birds, reptiles, mammals, birds, etc. But we also do find much more diversity in the fossil record. We find much more diversity in the pre-flood world. We find creatures that even seem to blend the features of 2 different types of animal groups. What we are looking at is a much larger design picture. Remember, the post-flood world is just a snapshot of the overall diversity that ever existed on the planet. The fossil record reveals incredible diversity. This means the pre-flood world was far more diverse than the post-flood world. Since what we are looking at today is only a snapshot of the entirety of biodiversity and of the pre and post flood world. Most of what evolutionists look to as transitional are simply pre and post flood diversity. With this amount of diversity to deal with, no wonder evolutionists love to line things up the way they want the evolutionary story to play out. When it comes to determining ancestry, what we need to consider is both discontinuities and continuities. With baraminology, the study of created kinds, you want to find a group that is discontinuous and separated out from every other group, and yet within it there exists continuity. This is a solid way to approximate the created kind. We find fossil groups that evolutionists point to as transitional-like creatures, such as the mammal-like reptiles with strong discontinuities. We do not find one continuous, unbroken chain from one type of animal to another. We find continuities within groups we predict are related, and discontinuities outside the groups. We find clear discontinuities in fossil groups. This means we have both fossil and genetic related data that demonstrates independent origins. We will be getting into the genetic data a little later on in this book. Let us look at the biological structure of life. We know that human engineers also build in nested hierarchical patterns. These types of groups within groups patterns are found everywhere in the design world. In other words, we find an echo of the overall pattern of life in the design realm. We can see hierarchical patterns at the visible level and the blueprint level. As biblical creationists, we would expect a human and a chimpanzee to share more in common in terms of overall similarity. Similarity in morphology, anatomy, physiology, and genetics than a human and a dog or a human and a fish. Even before we had genetic sequencing capabilities, you could simply stand back and predict more similarity in terms of genetics with a human and a chimpanzee. Of course, humans would have to be more similar to some creatures and less similar to other creatures. The next time you take your children to the zoo, have them pick out what they believe is the animal that most closely resembles humans. It wouldn't be long before they came back with a chimpanzee or a monkey. Biblical creationists do not need to fear similarity. Similarity simply demonstrates common design. As you can see in the above picture, figure 1, modes of transportation can easily be grouped into nested hierarchies. All sedans share a tremendous amount of features with each other. Sedans also share a lot of similarity with SUVs and vans. And although sedans have incredible similarity with SUVs and vans, as a whole, sedans still all share more with each other in terms of similarity. We can also bring in trucks, semi-tractor trailers, and even boats. And we would find different degrees of similarity, with sedans sharing more with SUVs than they do with semi-tractor trailers, and yet all still sharing more with each other than they do with airplanes or a tricycle. Evolutionary responses to these observations are typically very unsophisticated and are also based on obvious misrepresentations and emotion. A typical response I hear is cars don't reproduce. Adding reproduction adds complexity. It only makes the problem worse for evolution. And 2, this is a strong misrepresentation of our model of independent origins. Biblical creationists predict that if we are made in the image of God, as the Bible clearly tells us, then we should be able to get a sense for how God created the biological world based on how we as human designers build things. It is the observations seen in the design world that strongly helps confirm these expectations. The reason why critics of biblical creation will frequently misrepresent our position is because they are either not reading creationist material or they are reading creationist literature blindfolded. I also want to point out the fact that man is producing these vehicles for a purpose and for a function, an intent to move somebody from one place to another. God produces life, and he has given his creation the amazing ability to reproduce and pass on their genes. This is beyond anything man has ever been able to engineer. Cars, for example, are all made out of natural material. Iron, for example. This does not mean that cars made themselves. Humans, animals, and plants are made out of naturally occurring elements from the earth. Humans, animals, and plants did not make themselves in the same way that cars and other modes of transportation did not make themselves. Somebody made them from the natural occurring materials found in the earth. Man has dug up the materials from the earth. They melted it down and used the iron and steel. I'm simplifying, of course, for the point, but they use the metals to make cars. Man can do this. God can do this. But these materials cannot do this themselves. This is the point of the design model. As I explained earlier, from the design starting point, we should hypothetically be able to get a sense for how God may have created the biological world simply by looking at the many ways we as humans design things. This is because we are told in Genesis that we are made in the image of God. The data strongly indicates these hierarchies are here by design. And yes, we can classify life into groups and categories based on anatomy, morphology, and genetics. And by definition, humans can be classified and grouped within these man-made classification systems. To say that humans should be equally apart from all forms of life as compared to more similar to some creatures, such as the great apes, and less similar to other creatures such as fish, is a horrible argument and also considers very little. Militant proponents of fish-to-fisherman evolution, such as Aaron Ra, proposes what is called the phylogeny challenge, seem to be suggesting that man was created without the DNA, RNA, and proteins that make up life, and ultimately code for the anatomy and morphology of living creatures. This type of design would not make sense, and it would also hinder scientific advancement in terms of study and research. This is especially true when it comes to healthcare in various medical fields, since, of course, similarities in DNA help with cures and medicine. The critics of the design hypothesis will also point to the variations in the biological world when it comes to design patterns and challenge special creation by asserting God should not have used so many different designs. Why not use the same design? Why use a variety of different designs and not just one basic design that works? A couple examples in the biological world that they would point to would be whales and sharks. Both live in the water, but whales are mammals and sharks are fish, or marsupial mammals and placental mammals. Different reproductive designs, and yet both mammals. Once again, this criticism fails to understand the design hypothesis that I have laid out. Human engineers also build and design things with incredible variation. And remember, this is coming from the mind of man, and the mind of man is finite and not infinite like God. Why do cars in North America have steering wheels on the left side, while places such as Great Britain and Australia have steering wheels on the right side? This is all variation in design. These design patterns all serve a purpose and a function in the same way a whale, which is a mammal, is built for purpose and function, while a shark, which is categorized as a fish, is built and designed for function and a purpose. Take a look at the variation in shoe designs, or even the variation in steering wheel designs. Variation in designs abound in the design world. What we see in man's design is a reflection of God's design patterns that we see everywhere in the biological world. The differentiating lines of evidence come down to function. We are going to discuss how function is key to determining ancestry more in later pages of this book. This is not a long process of evolution. These hierarchies are there by design and are there for functional purposes. In past books and even debates, I have personally engaged in nearly 100 formal and informal debates with no answer by the critic. I have pointed to human-specific genes, multifunctional and multi-purpose genes, differential gene expression, unique transcription factors, non-coding DNA and its many amazing functions not predicted by evolutionists, FOXP2 gene, signs and lines, long non-coding RNAs, genomic networks, orphan genes, functional junk DNA, and more. These all demonstrate independent origins and refute universal ancestry. Chapter 3: Adam and Eve, the first couple. The evidence for the literal Adam and Eve is so overwhelming that it's even challenging to pick out a couple lines of evidence to discuss here in this handbook on independent origins. The vast amount of evidence and data confirming separate ancestry and a literal Adam and Eve is exactly why I have written so many books on this topic. This includes very technical books written specifically to refute the best arguments put forth by the critics of biblical creation, as well as books specifically meant to be more basic and that are not really meant to address every single objection the critic could resort to. The best evidence is almost certainly the fact that we have actually discovered the first couple in genetics. For example, we have discovered both mitochondrial Eve and Y chromosome Adam. There is no disputing the fact that there is one single Y chromosome ancestor and one mitochondrial DNA ancestor. Every single Y chromosome on this planet is nearly identical, and we have very little variation in the Y chromosome. Our Y chromosome is also incredibly dissimilar to the chimpanzee Y chromosome, less than 35% dissimilar when we consider overall gene content and overall architecture. The evolutionary community were shocked at the massive differences between human and chimpanzee Y chromosomes. Proponents of human evolution have had to invoke all sorts of rescue devices and hypotheses to explain the incredible discrepancy. We know that the Y chromosome mutates a lot faster than ever predicted or expected by the evolutionary community. The number of DNA differences separating men from the Y chromosomal Adam sequence is only a few. And since there is incredibly low variation in the male Y chromosome, this chromosome must be young. You cannot have a rapidly changing Y chromosome with incredibly low variation, and at the same time, this chromosome is purported to be old. No. What we see in terms of this uniparentally inherited DNA compartment is exactly what we would expect to find if biblical creation were true. To be specific, the male Y chromosome has about 4,500 years worth of mutation accumulation, which means that our last Y chromosomal ancestor would actually be Noah and not Adam. This is fascinating news. By invoking a rapidly changing Y chromosome to explain the incredible dissimilarity in human and chimpanzee Y chromosomes only adds more problems for those who want to so desperately believe that humans evolved from ape-like ancestors. We should find some highly divergent Y chromosome sequences on the planet today, and yet we do not. All the data we have suggests that this male-specific chromosome is young. the data we have from the Y chromosome, strongly supporting separate ancestry, has even led to some amazing testable predictions made by Dr. Nathaniel Jeanson of Answers in Genesis. The mitochondrial DNA also has extremely low variation as well. Before I continue, I want to take a moment to go over some genetics basics to fully understand the importance of this evidence that points directly to our first parents, Adam and Eve. In genetics, the man passes down the Y chromosome, and the woman passes down the mitochondrial DNA. These are the uniparentally inherited DNA compartments. We can make some seriously good predictions using these DNA compartments. If the Bible's account of human origins is true, we should be able to look at the mitochondrial DNA and the Y chromosome and trace them back to two common ancestors, two individuals. The data we have did not have to be true if evolution were true, but this did have to be true if the Bible were true. The Bible predicted this. The evolutionary community did not predict the genetic data that suggests we came from just two people in the not so distant past, mitochondrial Eve and Y chromosome Noah, coined Y chromosome Adam by the evolutionary community. Focusing in on mitochondrial DNA, and specifically Eve, the mother of us all, the mitochondrial DNA turns out to be exactly what we would expect if the Genesis account of human origins were true. Remember, this was not predicted by the evolutionary community. There is every reason to believe that if ape-to-man evolution were true, there would be multiple mitochondrial lines, with many even being shared with the chimpanzee. Human and chimpanzee mitochondrial DNA sequences are immensely different. Remember, the evolutionists did not predict this. They have decided to retrofit the data into their evolutionary story. Proponents of universal common ancestry are constantly having to invoke storytelling and rescue devices. Again, there was every possible reason to have discovered that this mitochondrial DNA ancestor was not so unique. We can construct some very revealing family trees with the mitochondrial DNA. Additionally, we can develop some very intriguing family trees with the Y chromosome as well. I will discuss both. First off, mitochondrial trees grow in branching-like patterns. This is due to mutations, changes in the nucleotide sequence of DNA. Basically, we can reverse the clock back to the point where we would find Eve. We could also start with Eve. She would have children, and her children would have children. Every time a child is born, there is the chance that the child would have a mutation. And over time, due to mutations, the family tree gains more and more branches. The next step would consist of backing this process up to where we discover the woman, of whom we have all descended from. We know that the mitochondrial DNA mutates fast, and we know that there are only a few DNA differences separating any two people on the planet today. This all confirms that we have all descended from Eve, the mother of us all. Proponents of evolution cannot explain why there is such low genetic variation in the mitochondrial DNA, nor can they explain why there are such few DNA differences in this non-recombining DNA compartment. This is precisely why they do not like observed mutation rates, pedigree-based mutation rates, where we can measure mutation rates in the present, support the model of independent origins. And since there are only a few DNA differences in the mitochondrial DNA, with many generations since creation, we can still account for plenty of purifying selection. Let us now focus on a Y chromosome phylogenetic tree derived by Dr. Robert Carter. This is a tree, a Y chromosome phylogenetic tree, figure 2. Dr. Robert Carter took all the Y chromosomes and made a Y chromosome family tree. This tree is remarkable because what we are seeing is exactly what we would expect if separate ancestry and biblical creation were true. Visually, you will notice an explosion from a central point. This is an explosion essentially from Noah, the last Y chromosomal ancestor of us all. But the most important aspect of this tree, I want the reader to notice, is the length of the branches. This is all essentially a reflection of DNA differences, a reflection of mutations over time. What should be noticed is that when we focus on the length of the branches, we realize that this is an incredibly easy number of mutations to account for in the biblical time frame. This reflects far too mutations if deep time evolution were true. The reader will remember this was the same problem we covered when discussing the mitochondrial DNA. We are not looking at tens of thousands of mutations here. We are looking at just a few hundred mutations, as would be expected if the Bible's account of independent origins were true. This data did not have to be true, and yet it is. Evolutionists are constantly having to retrofit the data into their evolutionary story. They did not predict low genetic diversity in humans today. They did not predict the one Y chromosomal line, nor did they predict the one mitochondrial DNA line. One last thing to consider is the fact that the length of some of these branches indicate that some people can pick up more mutations in the exact same amount of time. It is difficult to look at the branch lengths and conclude a definitive amount of time that has passed. Therefore, the pattern and the overall data is what is so significant. Sources found in hard copy of book. Figure 1. An unrooted neighbor-joining phylogenetic tree of the Y chromosomes based on the Simon's Genome Diversity Project, SGDP. Data from Carter, Lee, and Sanford, 2018. SGDP attempted to sample from a wide range of peoples. The result is a tree that is a good representation of total worldwide Y chromosome diversity. Note the clear central starburst and the irregular branches that display more mutations than close kin. In this unrooted tree, branches are allowed to spread out naturally. The evolutionary root would be located midway along the A1 branch. Forcing a root at that point would produce the squared-off stair-step tree, perhaps more familiar to the majority of readers, with long spidery branches leading to a few rare African lineages. But this unrooted representation allows for a more natural reading of the data. This is a mitochondrial DNA phylogenetic tree. Figure 3, found in hard copy of book. This photo is also from Dr. Robert Carter. He took this from the 1,000 Genomes Project. This is a family tree of all the mitochondrial genomes in the world. Just like with the Y chromosome phylogenetic tree, I want the reader to notice the obvious and incredible pattern. This is typically where proponents of ape-to-man evolution close their eyes. This pattern is exactly what we would expect if biblical creation were true. We see an explosive pattern, explosive growth from a single person. In fact, there is history on this tree. We can see geography. For example, there are African-specific groups, European-specific groups, Asian-specific groups, and so on. Not only is this fascinating data that confirms separate ancestry, but it is also empirical. When we focus on the pattern, we quickly realize that this explosive growth starts from a center point. The Bible claims to be the history book of the universe. Genesis tells us God created two people, Adam and Eve. Humans were supernaturally created by God and made in his image. The Bible gives no indication that we are related to chimpanzees or any other form of life. We should be able to test these claims to modern scientific data. As you have seen in both the Y chromosome and the mitochondrial DNA, the claims made in Genesis pertaining to human origins have been confirmed by empirical scientific data. Modern science has discovered Noah being our last Y chromosomal ancestor, and Eve. In addition to the overall pattern, I want the reader to pay close attention to the branches and the length of them. As with the Y chromosome family tree, we see these branches have different lengths. For example, have a look at the haplogroup in Europe, the HVR haplogroup. These differences in branch lengths indicate that various people groups have happened to pick up twice as many mutations as their cousin. Just like we have seen with the Y chromosome DNA phylogenetics, this creates massive problems for the assumptions behind the evolutionary molecular clock. If people can pick up twice as many mutations as other people, then we clearly cannot make assumptions as to when humans and chimpanzees split from a hypothetical common ancestor. We cannot look to the genetic differences between humans and chimpanzees and use those differences to make assumptions as to when they split, resulting in a very slow, unobservable mutation rate. Remember, proponents of human evolution never question their basic assumptions pertaining to universal common ancestry. They assume that humans and chimpanzees are related through common ancestry. What we are looking at in this phylogenetic tree is essentially random mutations in populations over periods of time. It is important to remember that with both the Y chromosome and the mitochondrial DNA, we see a pattern that fits the biblical base model of ancestry. In regard to the mitochondrial DNA family tree in Figure 3, we see evidence for one woman who lived recently. And so once again, and most importantly, this tree is an obvious reflection of only thousands of years as compared to hundreds of thousands of years, as the proponents of human evolution would have everybody believe. These family trees do not have a lot of mutations, and without any preconceived ideas about ape to man evolution and evolutionary molecular clocks, riddled with assumptions, the clear conclusion is that this tree and the Y chromosome tree in Figure 2 is young. Remember, this is one female ancestor from who we have all descended from, and she is nothing like the chimpanzee. Once again, independent origins is confirmed. References and sources for further reading available in hard copy of book. Chapter 4, The True Origin of Species. There is a study that has revealed through the genetic diversity in the CO1 gene of the mitochondria that over 90% of all animals came into existence at the exact same time. This is clear evidence for the global flood bottleneck. In a nutshell, what they discovered are clear genetic boundaries in species, which is exactly what we would expect to find if separate ancestry were true and not universal ancestry. When we consider this amazing evidence, Adam and Eve, the first couple, pertaining to the genetic data, confirming a literal Adam and Eve and separate ancestry, we see how amazingly consistent the Genesis account of origins is with the scientific data. Once again, evolutionists never predicted any of this data. The number of species on the planet today are perfectly in line with the young Earth creation timeframe. All we have to do is look at the number of species today on the planet and make predictions on the numbers of species, say within a family, that we should expect if the flood bottleneck occurred just 4,500 years ago. Dr. Nathaniel Jeanson of Answers in Genesis has done exactly this. I will use one example, but we could essentially go through all kinds of animals. Let us look at bird species. We know there are about 12 to 18,000 bird species on the planet today. Therefore, we would predict roughly 2 to 3 new bird species per year since the flood. Dr. Nathaniel Jeanson has made this prediction. What's amazing is the fact that we now have novel evidence for bird speciation events in real time, strongly indicating that there are far too few bird species today if deep time evolution were true. All numbers line up with the young Earth creation model of speciation. Remember, proponents of evolutionary theory invoke mutations as the source for all genetic diversity, while biblical creationists invoke created nuclear heterozygosity, millions of DNA differences in chromosome pairs. Therefore, all that needs to occur for rapid speciation events, since the DNA differences are already built in at creation, is recombination, gene conversion, migration, isolation, and shifts from heterozygosity, more allelic diversity, to homozygosity, less allelic diversity. Evolutionists require thousands of years for speciation events, simply because mutations need to add the necessary genetic diversity for speciation to occur. Take the bear kind that was brought on board the ark. The common bear ancestor of all bear species today would have originally been front loaded with pre existing DNA differences, which would have ultimately led to the origin of all the bear species, both extinct and alive today. God would have most likely sent the most heterozygous pair of bears onto the ark in order to allow for optimal variation and adaptation post flood. The same thing would also apply to the cat ancestor that was brought on board the ark. It is the young Earth creationists that are making the future testable predictions on speciation rates, some proving to already be successful. Proponents of universal common descent need to essentially explain the origin of all DNA diversity and how it is possible for genetic mistakes over time to accrue so much beneficial variation for natural selection to act upon. We know that mutations are the destroyer of genetic information and genetic complexity. Mutations degrade genetic functionality. It is much easier to break something down than to build something up. Mutations have been demonstrated to produce negative variation and not useful or beneficial variation. Natural selection is a fine tuning mechanism that keeps the species as strong as it can be. Natural selection works beautifully in the biblical model of ancestry. With the genetic diversity front loaded into animals at creation, mechanisms of change can act upon that useful diversity to create variation and also lead to adaptive episodes and events. When it comes to bird species, proponents of universal common ancestry say birds evolved from theropod like dinosaurs millions and millions of years ago. Let's say 100 to 300 million years ago. According to the numbers and the rates of speciation, we should be seeing easily 500,000 bird species today, and not the 12 to 18,000 bird species if deep time evolution and universal common descent were indeed true. The number of species and the rates of speciation events and the direction of genetic change are all contrary to evolutionary theory. We also have overwhelming evidence for forward thinking in genetics. This is another direct expectation of the designed diversity hypothesis. For example, we know we have our genes that make physical parts of our cells, while also having what is called the epigenetic inheritance. This is a whole additional layer of complexity and functional information that evolutionary mechanisms cannot account for. The epigenome controls how the genes make the physical parts of our cells. And this extra layer of amazing information helps turn genes on and off and not change the actual genetic code itself. Our genes are not all turned on at the same time. This would not be good. These are non random changes. Good design means we have to have some type of control, a type of control that makes sure we have the right things being turned on and the right things being turned off. This is all forward thinking, which points us back to the forward thinker. Remember, natural selection sees the short term. Therefore, how can evolutionary mechanisms build that which requires long term forward thinking? How does evolution build a spare tire? A spare tire requires the ability to think ahead. The evolutionary community never predicted such phenomenal rapid change and adaptation, all based on the pre existing capacity to vary and adapt. This all makes sense from the biblical creation starting point. Ever changing environments require ever changing genomes. In our amazing God, front loaded living organisms with the ability to change and adapt rapidly if necessary. These changes we observe are no help to large scale, pond scum to people type of evolution. These changes are not actually adding any real novel and meaningful genetic information. The information was already there, and the capacity for change already existed. What we observe in terms of genetic change is either functionally compromising to living organisms or general organismal adaptation based on the pre existing ability to change. This is why it is especially important to define terms when discussing these important topics with a proponent of universal common ancestry. Defenders of ape to man evolution like to hijack the word evolution to imply that change over time means fish and pine trees are related over time. This could not be further from the truth. Both creationists and evolutionists believe in change. We see change. The problem for those that accept universal common ancestry, the type of change observed and recorded is not the type of change that is going to drive large scale evolution that would take a single celled like ancestor billions of years ago into a whale or a human. In light of the information provided so far in regards to speciation and biblical creation related predictions, it now should be apparent that misunderstanding the model of those you wish to critique results in a ripple effect of bad arguments. This whole question, where does genetic diversity come from, is so incredibly important when it comes to the origins debate, simply because both models have vastly different views and very different explanations for the origin of genetic diversity and DNA differences. In the nuclear DNA specifically, when it comes to DNA differences. As we have learned, the proponents of universal common ancestry explain the origin of genetic diversity as the result of mutations over time, while the biblical creationist explains the vast majority of nuclear DNA differences as the result of pre existing nuclear heterozygosity. When the critics of biblical creation argue against young earth creation speciation by arguing against evolutionary creation, they are guilty of a straw man argument. Just by this one difference in terms of genetics, we have massive implications for the timing as to the origin of species. If the biblical creationists explain the origin of all DNA differences as the result of mutations over time, then they would be correct when they assert that the young earth creation model cannot explain the origin of all species in just a few thousand years. We, as biblical creationists, we're actually looking to mutations as the source for all genetic variety, then it would indeed be impossible to explain all the species we see today in just a few thousand years since the global flood of Noah. When you explain the origin of all DNA variety as a result of mutations over time, you require millions of years for all those differences to accumulate. Therefore, I am reiterating this important point, since when we start with pre-existing DNA differences at the start of creation, simple genetic processes such as recombination and gene conversion will yield visible variety in a single generation. We touched on this earlier in the chapter. Because evolutionists reject the design diversity hypothesis, We covered why they have no explanation for the origin of life in the first chapter of this book. They are incapable of considering this amazing explanation for the origin of species. This starting point leads to successful, testable predictions on speciation rates and DNA function. The design diversity hypothesis is exactly how we can get millions of species with incredible variation in just a few thousand years. It is only those that hold to universal common ancestry that require millions of years to explain the origin of species. We can also see the echo of design diversity in the breeds we have today. For example, we have over 700 breeds of horses and donkeys in the world today that both creationists and evolutionists would agree originated during human history, since humans are the ones that are artificially selecting for the traits that make each breed unique. Humans are the ones responsible for producing the hundreds of horse breeds we have today, indicating that all these breeds would have originated from a common ancestor at some point during human history. Animal breeds, for example, horses, dogs, etc., demonstrate that we can get phenomenal variety from pre-existing genetic differences. We only have 7 living equid species today. If we can get hundreds of horse breeds during human history that both creationists and evolutionists would agree with, then it is not very difficult to get 7 equid species, all from a more heterozygous pair of horses on the ark. When a critic of biblical creation asserts that creationists cannot account for a large number of species today in just a few thousand years, make sure to let them know what incredible variety we have produced on the farm. Chapter 5 of the Independent Origins Handbook: A Flood of Evidence. When proponents of universal common ancestry are cornered, they love to hang out in the fossil record. I discussed in detail earlier in this book the so-called transitional forms that evolutionists point to as evidence for their theory, and why essentially these types of mosaics are expected given the design model. Remember, a few interesting mosaics are no problem for independent origins. If fish to fisherman evolution were actually true, there would be countless transitional forms in the fossil record. What we find are examples of good design perfectly consistent with the design hypothesis, since human engineers build transitional like vehicles, amphibious assault vehicles, crossover SUVs. The best way to determine ancestry is through genetics. Genetics are inherited sperm and egg, not a fossil. We also know that there often exists more variation within the same species than between species. If every single domestic dog drowned in a flood and managed to fossilize and were dug up thousands of years later, scientists would make all sorts of erroneous conclusions, simply based on the incredible variation observed in the domestic dog. Without the genetics of a species, it also becomes difficult to tell what is due to homology and what is the result of convergent evolution, the independent evolution of the same traits. Anytime convergent evolution is invoked, it is an admittance into the lack of uniqueness of the universal phylogenetic tree. A good way to determine what is related and what is not is through breeding tests. It is difficult to determine gene flow based on fossils. Another thing to note that even if the proponents of evolution want to focus on this type of low quality science, since fossils are open to a wide range of storytelling and interpretations. We know from earlier in this book that creation scientists are discovering strong discontinuities in extinct fossil groups. As I have stated earlier, and I will reiterate here, we have the genetic data and the fossil record data in support of independent origins. The fossil record is also overwhelming evidence for the global flood of Noah. The critics of biblical creation scoff at the idea of Genesis being a real historical account of origins, when in fact the Genesis flood is the best explanation of what we see around us. Present geological processes cannot explain the geology of the earth. It is quite funny that these scoffers will look to Mars and yet have no problem talking about global floods that have apparently occurred on the planet. But they refuse to admit global flooding on Earth, which is absolutely covered by water. Mars is not covered by water. If the mountains today were flattened down and the ocean bottoms raised and everything was completely smoothed out, the waters of the earth would cover the entire planet in many miles worth of water. The critics constantly ask, well, how would the floodwaters have covered Mount Everest? There was no Mount Everest before the flood. These mountains uplifted extremely rapidly as a result of the flood itself. Meters per second movements of the plates. Remember from earlier, the critics typically use straw man arguments because they are not really up to date on creationist literature. The critics will assert that there is no evidence for the flood. This is laughable. All you have across entire continents. The cause of these sedimentary layers must be global in order to explain these incredibly massive layers extending entire continents. This all had to have formed rapidly. If these layers were not formed rapidly, we would not have billions of fossils in the first place. Fossilization is an exceedingly rare event and requires extremely specific conditions. To get a fossil in the first place, you have to bury it quickly and deeply. Because if an organism is not buried quickly and deeply, you will get scavengers who will eat the organism, or the organism will bloat and float, never actually becoming a fossil. It will fall apart and disintegrate. As always with evolutionary theory and even abiogenesis, time is not the hero of the plot. It is actually the enemy. Billions of years will not help with fossilization, since fossilization requires rapid burial. This must happen quickly, as would happen in a global flood event, as spoken of in Genesis. Once again, the reality and truth of Genesis is confirmed through scientific observations. We see massive dinosaur graveyards where we know this must have occurred quickly through catastrophic means. We literally have fossils of sea creatures giving birth and even fossils of sea creatures being eaten. Sea creatures do not lie on the bottom of the ocean taking millions of years to give birth or finish eating. This is the absurdity of uniformitarian and evolutionary thinking. Evolutionists have great faith. We have plenty of evidence for fossilized footprints, which again means there must have been truly little time before the next layer was laid down in order to get the fossilized footprint to begin with. The Genesis flood event would explain the necessary conditions for these rapidly formed layers. This is all fascinating evidence for sudden and rapid burial. The global flood of Noah cannot be denied. In some of the global flood models, we have runaway subduction where one tectonic plate goes beneath the other. If this were done over millions of years, the subducted plate down under the earth would have warmed up to the surrounding mantle rock. The Bible tells us that at the beginning of the flood, the fountains of the great deep were broken forth. This would suggest that most of the water of the flood came from below the earth's crust, in subterranean chambers circling the entire planet. Since the Bible clearly tells us that the fountains of the great deep broke open, we can make testable predictions as a way to actually test whether this really did occur just thousands of years ago. These fountains erupted in such a way that the original crust of the earth, due to the tremendous force, would have been cracked or fractured like an eggshell. We should find evidence for this cracking of the earth today. We find exactly what would be expected with the mid-oceanic ridge. This is very well known in the Atlantic ridge. If you follow this around the entire globe, much like a seam on a baseball, you find that this can best be explained by the original fracturing of the earth during the bursting of the fountains of the great deep. We know the plates are moving today through observations. The continents are moving very slowly. The catastrophic processes during the flood can explain what today's observed snail-paced geological movements cannot. When it comes to the global flood, we are no longer looking at the continental drift we see today. We are looking at continental sprint. It was predicted by a creation scientist that if this runaway subduction I was discussing earlier had only occurred just thousands of years ago during the flood, we should find evidence of this in the earth. And we have discovered this exact evidence. We have discovered huge slabs of cold rock at the base of the mantle, indicating that they had not yet had time to warm up to the surrounding materials. This means they have been placed there very quickly, 4,500 years ago as compared to millions of years ago. If these slabs were placed there millions of years ago, they would have warmed up to the surrounding material or even melted. If these plates have descended into the deep hot mantle, these enormous slabs of cool rock discovered should not have been cool as predicted by flood geology. Chapter 6 of the Independent Origins audiobook, The Stronger Model. The reason why the evidence best supports the independent origins model is because numerous lines of evidence exist that only the separate ancestry model can explain. This model also makes the best testable predictions, as we have covered throughout this book, which is the gold standard of science. It was the evolutionary community that set this standard, and it is the creationists that are meeting this standard. From the design diversity starting point, the biblical creation model predicts that the vast majority of DNA sequences and DNA elements are functional. We have preliminary evidence for genome-wide functionality in humans. Universal common ancestry requires genomes of junk, genomic fossils, and evolutionary leftovers. Intelligent design advocates have always predicted that we would discover important functions for non-coding DNA. And these DNA function-related predictions have shown to be successful. We know that the vast majority of our genome has incredible evidence for biochemical function. A lot of this data is preliminary, which is why creation scientists are able to look at this data that suggests function and make future testable predictions. But with all this evidence for activity, why else would the cell be so active? If this activity was not useful, or the transcription was simply spurious, then natural selection should have weeded out this genetic baggage millions of years ago. This would be incredibly wasteful of resources and energy for the cell. This is a strong indication that this activity is in fact useful and beneficial. Evolutionists have also looked to pseudogenes as evidence for ape-to-man evolution. This line of evidence, shared genetic mistakes between humans and chimpanzees, has also been overturned. Pseudogenes have been shown to be essential and necessary to sustain healthy life processes in the cell. The evolutionary community did not predict the extent to which the genomes of living organisms were functional. The evolutionary community has looked to junk DNA and have invoked an argument against the design model. They used to assert, and many still do, that over 90% of the human genome was evolutionary junk. If the human genome really was mostly junk, then the critics of biblical creation would have a point. Intelligent agents design things with function and purpose. If most of our genome turned out to be useless genetic baggage, then this would be a solid argument against the design hypothesis. As I iterated earlier, this argument has been demolished. Proponents of evolution arguing against biblical creation have been guilty of materialism of the gaps with their line of reasoning and argumentation. Their assumption that most of the genome was genetic baggage was based on an evolutionary starting point. Evolutionists require neutral junk in order that deleterious mutations could be absorbed by the junk areas of genomes, making them absolutely neutral and unharmful to organisms and genetic functionality. In 2012, the ENCODE project came out with amazing results, suggesting that upwards of 80% of the human genome was active to some extent. Detectable biochemical function. We have seen an overwhelming number of papers come out since then, suggesting more and more function for our genome. Papers are always coming out identifying specific functions for genetic sequences once thought or assumed to be useless. Junk DNA could have falsified the design hypothesis, and yet it turned out to be completely wrong. We have fascinating evidence for function in endogenous retroviruses and even in ALU sequences. Proponents of evolution have claimed that endogenous retroviruses were the ancient remnants of viral infections and strongly confirm evolution. The real question should be, are these really the ancient leftover remnants of viral infections, or are they created units of DNA function? They have also lost this important piece of evidence for common descent, given the incoming evidence for DNA function and design. The evidence we have for DNA function also makes large-scale evolution that much more implausible, simply because the mutations that are accumulating from generation to generation in living organisms are that much more deleterious. The more functional the genomes of living organisms are, the more quickly deleterious mutations will degenerate genetic information and genetic functionality. Evolutionists have a major unsolvable problem for mutation accumulation and genetic degeneration. Mutations are accumulating far too quickly for natural selection to filter out the damage. Defenders of common descent have failed at every opportunity to present a type of selection that can filter out so many mutations accumulating from generation to generation. Most of these mutations are nearly neutral and therefore invisible to selection. These nearly neutral mutations are subject to drift, making selection ill-equipped to stop genetic degeneration. Another major problem for forward evolution is the fact that most so-called beneficial mutations are reductive and functionally compromising to the organism. For example, sickle cell anemia is a commonly repeated example of a beneficial mutation, and yet it is due to something that has been broken. This beneficial mutation may have an advantage, but overall it is not taking things forward. It is reductive. A broken hemoglobin protein is not going to solve the problem of genetic degeneration, nor is it going to drive an evolutionary process that takes a single-celled like ancestor into a whale over time. Defenders of evolution require a way to counterbalance the damage done by the large number of low-impact mutations pouring into the genomes of living organisms. Large-scale evolution would require massive numbers of truly beneficial mutations to counterbalance the net loss of genetic functionality. Compensating for loss of genetic information and functionality is just not possible. There is a relentless influx of deleterious mutations that are pouring into populations over time, and these are mostly invisible to selection. Proponents of evolution do not have a solution to this major problem that is extremely powerful evidence against universal common ancestry. When we see adaptive events due to mutations, what we are looking at is a change only beneficial in a linear sense, since overall incredible numbers of low impact mutations are accumulating. These deleterious mutations are eroding genetic information. Apologists of evolution will often suggest that there is a trade off, when in fact the trade off is not sustainable. This is because while you may be improving a few nucleotide sites, you have large numbers that are degenerating in many other ways. As we have seen with bacteria, this often leads to shrinking functional genome sizes. The human mutation rate is roughly 100 new mutations per person per generation, with most of these being unselectable. The damage is mostly invisible, as it is much like rust on a car. You cannot exactly see each rust event on your vehicle, but over time, regardless of how many times you bring your vehicle in for servicing, it will rust out. You can also think of it a lot like a single spelling mistake in a book the size of an encyclopedia. On its own, a single spelling mistake does very little. But over time, when enough spelling mistakes have accumulated, the book becomes unreadable. Genetic degeneration points us back to the perfect creation of Adam and Eve, where there would have been no mutations. This would have been a time of perfection and increased lifespan. Genesis tells us about the fall of man, which would predict death, degeneration, and extinction. This is exactly what we see. Genesis also records a time where people lived to extremely long ages. The reality of genetic entropy reminds us exactly how this was possible. Humans once lived hundreds of years old. And because of mutation accumulation, we have progressively declined to what we see today. It is also important to note that other factors would have also reduced life expectancy. For example, harsh environmental conditions post flood would have resulted in a decrease in longevity. The pre flood environment was much more pure, clean, and suitable for long lifespans and exceptional health. Our hope is in Christ, and the ripple effect of the fall reminds us of our need for the gospel. One day he shall wipe away all tears, and death shall be defeated. The reality of genetic deterioration is sad, but we know Jesus Christ, our Savior, has promised to restore his people to a glorious state. There shall be a new heaven and a new earth. And if you are not yet born again, please consider what Jesus has done for you in order that you too can have eternal life. The Independent Origins Handbook, audiobook. Conclusion. The evidence for special creation and independent origins is overwhelming. We have also seen throughout this handbook that the scientific data strongly corroborates the validity of the account of origins as recorded in Genesis. We are told in the last days, scoffers would be ignorant of the global flood. And even though the scientific evidence demonstrates sufficiently that the worldwide flood of Noah was a real event, the opponents of biblical creation continue to assert that there is no evidence for this worldwide event that shaped the earth into what it is today. To reject Genesis as literal history is to deny the data. Take advantage of this book by looking to it as a comprehensible resource that presents a compelling case for biblical creation, as well as a powerful refutation of universal common ancestry. This is all accomplished in an approach that is both readable and understandable to both non-scientists and experts, as a way for it to be easily accessible anytime an answer is needed concerning the origins debate. Appendix: Created heterozygosity model explained by Ramat. If we look at the nuclear DNA and mitochondrial DNA as a battery, then it helps us to visualize our model in a very helpful and beneficial way. Different kinds of batteries represent different related kinds. The charge of the battery determines mutation rates over time. And so over time, the loss of charge is analogous to mutations building up over time. And as more time goes on, the battery loses more and more energy. If evolution were true, the battery would be totally out of charge since they cannot last forever. What do we see? We see that mitochondrial DNA has not even remotely accumulated the amount of mutations that should be visible if deep time evolution were true. In other words, the batteries in all life are nearly fully charged, yet draining quickly. What does this mean? This tells us that not a lot of time has passed. It also tells us that if batteries are losing their charge quickly over a short period of time, then we can make predictions based on this. As Donnie has covered in this book, testable predictions are the gold standard of science. Which model better explains the data is ultimately going to come down to who is making the accurate testable predictions. When we look in the mitochondrial DNA and find more mutations, a battery not as fully charged, this represents more genetic diversity in that species. And those who believe in evolution equate it to being more ancient. Is this the case? Absolutely not. Since Noah's flood was a global bottleneck, and it reset the battery charge in all life forms worldwide, then we have no reason to assume that Africans with more mutations mean they are older than Europeans who happen to have less mutations, since they are all humans who descended from the same family on the ark. Therefore, we as young earth creationists have decided to make predictions based on different mutation rates in different human lineages, and also in different animal species, including those within the same family. Those that believe in evolution cannot make predictions on mutation rates because they admit that evolution is too unpredictable to make accurate predictions on. What they have just admitted is that they do not even meet the standard of a scientific theory, since they are unable to predict anything. Also, if you cannot observe it, test it, and repeat it in a lab, then it does not even qualify as scientific whatsoever. And all of these criteria match evolution down the line. What are you looking at is the difference between mitochondrial DNA and nuclear DNA. We use nuclear DNA to determine what is more related to one another, and mitochondrial DNA to determine age. This means we look at how fast mutations are arising and then look at how many have accumulated, including those that have reached fixation, which determines how old the creature is. Using this criteria, we can clearly see that all life is extremely young, and the global flood bottleneck must have been recent. We at Standing for Truth Ministries have directly made many predictions based on young earth creation, and you can find this research yourself by searching for the study titled Invalidation of the Evolutionary Molecular Clock, Neutral Theory. You can find it on our website, creationistclothing.com, and our official website, standingfortruthministries.com, under articles and also on ResearchGate.